So here we got a negative correlation between number of resistant genes and disease severity.
Yeah.
No?
Yeah.
Okay, but I want to know whether there is any correlation between yield potentiality and number of resistant genes.
Well, intuitively, there would be, right? The question is whether there's a relationship between the number of resistant genes and yield protection. Okay? Robert showed us a case where he had many genes and a very strange response. One problem is that at this point we really don't know how many genes are in the varieties. We're guessing right here. This estimate that Franklin only has 1 to 2 minor genes is based on some adult plant data from a field evaluation in 2008. It was scored on 2 different occasions as about a 15 MRMS, something like that. Donda had a much better score, MSMR. Kingbird had a 0R, 5R, 5MR, very, very different resistance. And we hypothesized that amount of resistance was a function of the number of genes that each variety had. We knew that they did not have any major all-stage seedling genes. So, getting to the conclusions again. So is the problem with the way the data are presented, or is it, you still have a question about the presentation or the results?
This conclusion is expected.
Yeah, I think so. This is what we thought. With protection, this is what we thought would have the highest yield gain, and this is what we observed. And in fact, the ranks are exactly what we thought they should be. And I think without the benefit of the statistics I should have to help properly interpret these numbers, I'm suggesting that there is no difference in the protection between Kingbird and Eagle 10. There is no benefit at all to applying a fungicide.
Yes, you are right.
And I think these, I would have to be a very poor researcher to have a magnitude of difference like that and not be able to declare it to be statistically significant. This is a little, I wish I had the precision to help answer my question here. But I think that is probably significantly different from 0. And I think that is probably significantly different from 0. I'm just not sure if those are significantly different. And so looking at the ranks or the slope of the lines in response to fungicide treatments, I can only say that there appear to be statistically significant changes in rank between the APR varieties. So all APR varieties are not the same. We hope to get to a level of protection that that Kingbird has, and rely on that kind of multi-genic protection to avoid having to spray a fungicide.
The message from that slide is the varieties which contain APR don't need for fungicide.
The message is that some varieties with APR don't need a fungicide. This one is APR.
Yeah.
We think it has 4 or 5 genes. That's quite a few. These two, we're not so sure. This is Danda. We think it may have 2 or 3 genes, but I think, and again, I don't have the statistical, the appropriate statistics to correctly analyze or answer the question of, is that a significant response to the fungicides based on the precision I have in the experiment? I think it is, but I cannot tell you. And likewise, we think this one has only that many APR genes, and therefore its response to fungicide treatment protection was much higher. So all APR varieties are not the same. I think if you were in the field and you were evaluating kingbird for a number of years in a number of locations and you read it all the time, 5MR, 5MR-MS, 10MR, and even at near maturity, it's still reading very low, then I would be comfortable in saying, well, Ravi, you're right. This guy that's growing this APR variety, really, he should not spray. But in fact, in the absence of that information, I could not answer that man's question, and I said, I have no data. So.
Yes. Gordon, you were to make some points about the days to maturity plus 3. What was that?
Yeah, that's the difference in maturity. Danda is very late maturing. And I think, first of all, if you remember the table that I showed you about our fungicide scores, I think those estimates are underestimated because the terminal rust scores should have been, we would have liked to have had them be 0. So if this was 0, in other words, the 2 fungicide treatments had fully protected the check, or had fully protected the 2-gene variety, or had fully protected that variety, this should have been 0, 0, and 0. So there is some probability of at least some loss that was not detected because we didn't have enough fungicide treatments, or we didn't have enough fungicide, or we had the wrong fungicide. So I would suggest that the numbers that we ended up with at the end are low estimates. And
I mean, also, for me, it illustrates just how difficult it is to control stem rust with fungicide. I mean, your 2013 data is quite indicative. So with tilt, I mean, that's a common one that the farmers use.
Yeah.
And farmers would say, yeah, we have trouble controlling stem, tilt isn't working. And I suspect your data is showing, I mean, Rex Duo is a much more advanced chemistry. And that's, without a doubt, the best fungicide we have in Ethiopia, registered in Ethiopia. But even with that, and 2 sprays, you're still not getting complete control. And that's on a field controlled, it's not farmer field.
Dave knows pretty well that farmers are reluctant to spend money. There were recommendations last season that farmers apply a fungicide. The fungicide had been stockpiled by the government of Ethiopia. Farmers had been told to spray, warned of the danger. Some of them listened, many did not. A number of them had a harvest of 0 tons per hectare.
We should also keep in mind the application process. There's very little downward or translocation of fungicides from the leaves to the stems. So if you don't hit the stems at the bottom, you will have poor protection.
Yeah.
That message also needs to get out.
Rate of carrier of the water and
the penetration into the canopy.
Yeah.
Don't just spray the flag leaves or the upper part of the canopy. You've got to get inside. specifically for stem rust.
Yeah,